Hello, everyone. I'm very excited that you are all able to join us today. We have a fantastic speaker from Tulane University's National Primate Research Center who is going to share a revolutionary way to work with infectious materials. My name is Matt Riddle, and I am Miltenyi's flow cytometry specialist based out of Houston, Texas. Before I introduce our speaker, I'm going to give a brief introduction into the platform that she is going to be discussing, the MACSQuant Tyto Cell Sorter. I will describe the way that the sorting has been done in the past, the problems that occur, and what makes a Tyto unique from a biosafety perspective to solve these problems. After my overview, you will hear about how this instrument is revolutionizing cell sorting in the BSL-3 environment. Without further ado, I'd like to introduce the MACSQuant Tyto. It is a closed and self-contained benchtop cell sorter. It is quite compact and it does not require any fluidics, which eliminates sheath fluid or waste containers that need to be stored underneath. The large external monitor makes it easy to analyze data and implement a gating strategy. A disposable sort cartridge holds the sample and is housed in a closed and temperature-controlled chamber where the sample can be kept at a constant temperature from 4 to 25 degrees Celsius. Tyto has 3 lasers, one violet 405 nanometer, one blue 488 nanometer, and one red 635 nanometer. There are 8 fluorescent channels, 2 off the violet, 4 off the blue, and 2 off the red, plus a backscatter off of all 3 lasers and side scatter off the blue laser. You can find more information of the Tyto specs on the Miltenyi Biotec website. The Tyto is capable of sorting many complex phenotypes, including, but not limited to T cells, NK cells, Tregs, B cells, microglia, yeast, bacteria, iPSC cells, and nuclei. And yes, you did hear right. Laboratories are currently sorting yeast, bacteria, and nuclei with the MACSQuant Tyto. As most of you may know, sorting is traditionally done in what we call a droplet-based method, as you see here. During this droplet method, cells can be subjected to high pressures and shear forces. The cells are centered in a flow using sheath fluid. They then enter the flow cell where they are interrogated by the lasers and proportioned into droplets. They are then subjected to a charge to direct them into the appropriate collection tubes below. During this process, the high pressure and open nature of the droplet-based sorter can create aerosols which can be harmful to the user by putting potentially dangerous substances in the air. Today, you will learn about how the flow cytometry core at the Tulane National Primate Research Center tested the MACSQuant Tyto for use in their BSL-3 facility. But first, you need to understand how this flow sorter is different from a droplet sorter and how Miltenyi revolutionized the sorting process with a result that significantly lowers the risk to the users. At the heart of this solution is the MACSQuant Tyto cartridge. It has 3 chambers: the input chamber, the positive chamber in the middle, and the negative chamber. All chambers can be accessed from above through allure lock ports. Different from the conventional droplet sorters, the MACSQuant Tyto has no fluidic paths inside the instrument. In other words, the cells do not run through the instrument itself. Instead, prior to the sort, cells are loaded into the cartridge and remain in the cartridge throughout the entire sort. Your sample remains in an aseptic environment until it is removed after the sort is complete. To maintain this sterile environment of the disposable cartridge, each of the 3 chamber access points are covered with a allure lock cap, and all ventilation ports are covered with a 0.1 micron hydrophobic air filter. This is especially important when sorting in hazardous samples. To maintain an aseptic environment and increase the user safety, the cartridge can be loaded and unloaded under a hood. Here, you can see the different chambers of them along with the microchip, which contains the sorting valve. You'll see the input on the left, the positive in the middle, and the negative on the right. Compared to the droplet style sorting, this sorting valve is how the Tyto isolates your cell of interest. With a low pressure flow, your sample will move from the input chamber through the microchip where it is interrogated by the lasers. After these lasers is where the valve sits. Like a door, the valve opens and closes to control the direction in which the sample flows. Both the negative and positive populations are kept in the cartridge until removed by the user under a sterile hood. At this point, the sample can be taken for further analysis. Also, the negative population can be collected and sorted again. This process is best shown through an animated video. First, you will see where the light air pressure enters the cartridge to create the flow for your sample. This is where it will pass through at the first 0.1 micron filter. This starts the mixing propeller to keep your sample suspended. Once you start your sort, the cells move from the input chamber through to the microchip to either the positive or the negative chamber. You can see here how they pass through the lasers and the valve opens and closes to control the flow of the green target cells to the positive chamber and the other cells into the negative chamber. Now that you've seen the instrument and how it works, I'm excited to introduce our featured speaker, Dr. Marissa Fahlberg. She's Assistant Director of the Flow Cytometry Core at the Tulane National Primate Research Center. As an early adopter of the Tyto technology, Dr. Fahlberg is one of the most experienced Tyto users in the United States. She will discuss some of the testing that was done in order to demonstrate the safety of the instrument while sorting hazardous materials.
Thank you for the introduction, Matt, and thank you to everyone for joining this webinar. Today, I will be talking about our journey and experiences validating the Tyto for BSL-3 sorting. My name is Marissa. I am the Assistant Director of the Flow Cytometry Core at the Tulane Primate Research Center, which is located in Covington, Louisiana. It's about an hour north of New Orleans. We primarily focus on improving human health outcomes from infectious diseases. The flow core is housed within the Division of Immunology, and we have both analyzers and sorting services. We support our internal investigators as well as external collaborators and NIH contracts, and we're always welcome to work with anyone. So if you're interested in what we do and what we show you today, then feel free to reach out. So we have a lot of demand for sorting at the Primate Center, both BSL-2 and BSL-3 pathogens, as well as select agents, which includes tuberculosis, SARS-CoV-2, Coxiella burnetii, which causes Q fever, Rickettsia parkeri, anthrax, ricin, SIV. SIV is the simian or monkey homologue of HIV. And a lot of times, if someone wants to sort a pathogen that's typically considered BSL-2, we will often sort it and consider it BSL-3 when it comes to sorting. That's because when pathogens are aerosolized, they can be more dangerous when you inhale them as a mode of transmission in comparison to a more natural route like ingestion or direct contact. For that reason, because aerosols are more dangerous when they're inhaled, and because self-sorting creates aerosols, conventional self-sorting is classified as a high-risk laboratory procedure. Aerosols during sorting are most often created during an event like a nozzle being clogged or obstructed, and even without a nozzle being clogged when high pressures are used, like 70 psi to sort. And 70 psi is a pretty standard pressure to use in combination with a 70 micron nozzle. 70 micron nozzles have a very steady stream in general, so it's a popular nozzle size, but it does come with a high pressure component to it, and so there is always the risk of generating aerosols in conventional self-sorting. So I wanted to show a video about the most hazardous aerosol production when a nozzle clogs. And this is a look inside of a conventional cell sorter. This is from a BD Fax ARIA Fusion. And at the top is the flow cell, and their nozzle is right here. It's a little white piece, and the stream flows through the nozzle between the deflection plates here and here, and down into this center piece here. And then from there, if all is going well, the stream will be deflected in 1 to 4 directions, sorting your desired cell types. However, when this nozzle clogs, and that can be due to various reasons, sticky cells like cancer cell lines or dead cells, or a sample that has a lot of junk or say mucus in it. If a nozzle becomes clogged and the stream deviates to the side here and doesn't go right through the middle, it hits the side, it will create this cloud or plume of aerosols that gets everywhere. And this is this is a home video, so it's not the best, but you can clearly see a ton of aerosols being created here, and droplets of all sizes going in, a million directions. And on top of that, when this happens, if you don't catch it right away and turn the stream off, sample can also go into the wrong collection tube, and that can contaminate your sample. And that is a real problem with conventional cell sorters, because anything that's not sorted is pushed into the waste. That sample is gone. And if you want to resort a sample because it's been contaminated, then you would have to spin everything down and resuspend and sort again. And now you're also pushing your sample through the sorter for a second time. So sequential sort, it's not the best for the cells. So it's a huge biosafety issue to have a situation like this, and it can result in sample loss. To mitigate the risk, the biohazard risk of a clog and the aerosol formation, there are several safety mechanisms that are in place and recommended by the industry. So one is to close this door. There's a physical barrier there. The second is to close that whole area. The third is to have your sorter inside a laminar flow biosafety cabinet. And the fourth is to use an aerosol management system, which evacuates aerosols from this area. And in combination, this system does work to vacate aerosols and keep the operator safe. The problem with the system is that it is cumbersome and it is time consuming, and it requires ideally that you test the aerosols on the same day as a sort. So sort days already are generally pretty long, and you have to set up the instrument, and that's for regular self-sorting. If you add the aerosol management component because you're sorting something that could be dangerous upon inhalation, it's going to add a substantial amount of time and difficulty. So an ideal system that's better than the current conventional system is one in which no aerosols are created. If that is not possible, then the second best would be a system in which some aerosols may be created, but they cannot escape, and you don't lose sample if the system fails. So we turn to the Tyto as a potential candidate cell sorter for RBSL3 to see if this instrument, which is a microfluidic cell sorter and has a completely different way of sorting compared to conventional cell sorters to see if it could solve these problems and bring us closer to the ideal solution or system that we want, which is essentially to create few or no aerosols and not allow them to escape. The way that the Tyto works, which is unique from a conventional cell sorter, is all of the sorting happens within a cartridge that's made of a hard plastic instead of going through tubes and a flow cell and nozzle. Everything happens inside this cartridge. So the instrument itself has no fluidics inside of it. The instrument has optics and electronics, but all of the sorting and fluidics happens within the cartridge, specifically in the chip, the microchip that is attached to the bottom of the cartridge. And air comes in through the input chamber. This is the input chamber where you load your sample. And the air pushes cells through the input chamber, pushes your sorted cells into this middle chamber or the positive collection chamber, and the cells that you're not interested in into the negative collection chamber. There is a 100 nanometer hydrophobic multi-layered filter above each of these chambers, which is required for the airflow. Within the input chamber, there is a mixing propeller that mixes at 800 RPMs. It is adjustable. The reason for it is to keep the cells in suspension. And then there's a 3D filter, it's called a 3D filter, that the cells have to pass through before they enter the microchip. And within the chip, there is a magnet. Well, the magnet is in the instrument. Within the chip, there's a valve that pushes the cells at an incredibly fast rate into the positive collection chamber or the negative collection chamber. Depending on the way that it's designed, there are several strengths as pertaining to biosafety for the Tyto in comparison to conventional cell sorters. So one is that the operation for a regular cartridge, because they just recently came out with a high-speed cartridge, the regular cartridge operates at about 3 psi. I think it's actually a little bit less on ours, and 17 psi for the high-speed cartridge. In comparison, I mentioned this earlier, a conventional cell sorter with a 70 micron nozzle will operate at 70 psi. And if you use a bigger nozzle, 100 micron nozzle, that's usually will sit between 20 and 23 psi, which is lower. In spite of that, even the high-speed Tyto cartridge is lower than that. So with the lower pressure, fewer aerosols are formed, status quo, just as it is. And then on top of that, the system is closed besides the filters, the 3 filters located at the top, which again are 100 nanometer pores, multi-layer, and they are hydrophobic. And there's no nozzle to clog. That's the big one. The clogging of the nozzle and that huge cloud of aerosols is the primary cause of aerosol formation in conventional sorter. Without a nozzle, that whole cloud of aerosols that you saw goes away. There is a filter, as I mentioned, that can be clogged, and that could potentially create a pressure issue, an increase of pressure into the input chamber if it's clogged and if the system is trying to push cells through. In that case, you could have a higher pressure situation, which could create some issues at the interface between the air and the liquid. But if the 3D filter is clogged, the other nice thing is that nothing goes to waste. The sample that you don't sort doesn't go into the waste. It just goes into this other chamber. So you never have to worry about sample loss on the Tyto, and that's one of my favorite things about it. If you were to have to sort again for some reason, it's true that you would have to put the sample through one more time, just like you would a regular sorter after you spin it down. But there's a big difference between putting a sample through twice at 3 psi or 17 psi compared to 70 psi. Some limitations of the system are the mixing propeller. It does mix at 800 RPM to keep the cells in suspension, and that could cause some splashing or aerosol formation potentially. As I mentioned, a partial or complete clog of the filter here could increase pressure on the system in order to try to drive and push the cells through. And then the 3 openings on the top of the chamber are the only parts of the cartridge which are exposed to the environment or to the air, and that's where aerosols could potentially escape if they are less than 100 nanometers in diameter. And they would still have to pass through multiple layers, and the filters being hydrophobic, would repel some droplets. But it is still possible because they are open to the environment. Just again. So we wanted to find out if what we thought was true about the Tyto, that it wasn't probably creating aerosols, or if it was, they were unlikely to escape because of the design. And so we set about determining whether aerosols were generated inside the cartridge, so within each of the 3 chambers. In addition, we wanted to investigate whether aerosols were formed outside of the 3 chambers and above those filters. And finally, we wanted to see whether any aerosols that were potentially generated outside of the cartridge, were they escaping the instrument above where the sort chamber door is closed, because typically you sort like this with this door being closed, everything is happening inside here. But there is a little space here where the door closes. It's pretty snug, but we just wanted to see if aerosols could escape through there, or alternatively, if they could escape through the bottom of the instrument here, because there is a vent system where air that's inside the instrument gets pushed out in this direction, left to right. And so we wanted to test this air here to see if aerosols could be in there. The industry standard for aerosol testing for conventional cell sorter, which has really been defined and optimized by Steve Perfetto at the NIH, is to create a worst case scenario artificial clog in the sorter nozzle, as I showed you in the video. And the way you do that artificially without actually clogging the nozzle is you just move the sort block over to the left a little bit so that the stream is hitting the side of where it's supposed to be, and creates that big cloud. And then you run fluorescent beads through the sorter at a very high event rate, about 40 to 50,000 events per second. The beads that are typically used are 1 micron in size, and that's because when you analyze a large number of aerosols that are created from, like what I showed you in the video, the concentration of beads at 1 micron in size tends to be higher than beads below that or higher than that. So it's thought that using beads that are 1 micron are a good representation of whether you have aerosols of any size. If you don't see the 1 micron, if you can't detect the aerosols that are 1 micron, then it's less likely that you'll detect aerosols smaller or bigger than that, if that makes sense. And then we use Cyclics D cassettes, which are these guys, to sample the air at various locations around the system. And these are very interesting. These are typically used for environmental sampling. And so it's perfect for this. What you do is you attach the cassette to one end of a tube, and then the tube is attached to this vacuum pump, which pulls in air at 20 liters per minute. So there's air being suctioned through this vacuum at 20 liters per minute. And you can sample various areas around the instrument to see if you can identify aerosols. So once the air gets pulled into this cartridge, into the cassette, inside the cassette, there is an adhesive that's also a mounting media. And the air gets pulled into the middle and sticks to that mounting media that's on a cover slip. The cover slip sits like right in here. And then you can just take the cover slip out and attach it to a slide and look for the beads under a microscope. And if you see beads, then that is indicative that aerosol was formed and you were able to recover it from the air by pulling it in through that vacuum. So that is the industry standard for aerosol testing for conventional cell sorter. We wanted to reproduce that industry standard for the Tyto as closely as we could, given that these are very different systems. So we created, or we tried to design this experiment where we would create a worst case scenario clog in the input chamber, clogging of that filter I mentioned earlier. We don't have a nozzle here. So whereas, in a fax aria or fusion, we would clog the nozzle, here we're clogging the filter in the input chamber. Secondly, we would run fluorescent beads, just like they do in a conventional sorter, except we ran beads that were 50 nanometers, 100 nanometers, and 1 micron in size, instead of only 1 micron. And that is because the filters, as I mentioned at the top of the cartridge here, these are 100 nanometers. So although for conventional cell sorter, the 1 micron bead is a great indicator of whether you're creating aerosols smaller or larger than that. In this case, the 100 nanometer filter is preventing beads that are 1 micron, most likely, from escaping. But it doesn't mean that aerosols aren't smaller than 100 nanometers or 100 nanometers are not escaping. So we couldn't just rely on the 1 micron beads when testing aerosols on the Tyto. So we also found a way to move the beads through at a very high event rate on the Tyto. And then we use those cassettes, the cyclic steed cassettes, to sample air at the different locations around the system and analyze those samples under microscope. Initially, we had to do several optimizations to get the testing in place. First, we had to determine if we could even detect these tiny beads under a microscope, because 50 nanometers is incredibly small. And then we had to develop a method to artificially clog the system, because it wasn't as easy as just moving a sort block over and creating the cloud. We had to actually clog that 3D filter. And then we needed to determine the sample volume required to maximize spray potentially from that mixing propeller. So this is just showing that we were able to identify the beads at 50 nanometers, 100 nanometers, and 1000 nanometers. These are different magnifications. And the 50 nanometer beads, I'm just pointing this out, they sometimes look a little bigger than the 100 nanometer beads. That's not an error, that is aggregation. So although there were a lot of beads that were by themselves, so the beads were still good to use for this test, they were stickier. The 50 nanometer beads were stickier than the other sizes, but we could see them all. The clogging efforts is where things got a little bit tricky. The system was very, very difficult to clog, which is great for a sorter, but it is challenging when you're trying to do it on purpose for aerosol test. So we tested 10 micron beads, which is what you see here. We tested trying to clog the system with undiluted whole blood, with whole blood mixed from several animals in order to activate the cells and try to get them to be stickier, both same day and overnight mixing. We tested homogenized brain samples with and without the junk that you get from digesting the brain from a monkey in this case. We tested cell lines, we tested thymocytes and splenocytes that had a lot of dead cells in them. We got desperate, and so we tested Elmer's glue at increasing concentrations, and we even tested agar and tried to dry some agar on top of the filter at increasing concentrations. We were able to clog the system. Basically, we had two outcomes. So we either completely clogged the system, in which case the pressure actually dropped every time consistently from 120 HPa to 50 HPa, which is about 0.73 psi. Instead of the pressure being greatly increased, it just dropped time after time. The other alternative was we were sometimes able to get the pressure to go from 120 to 150 HPa at the maximum, but that's a difference of going from like 2.5 psi to I think like 3 or 4 or 5 maybe psi, which is really nothing. We could never get the system to get to a really high pressure. And here you can see this is data from the B testing, where the more the filter was clogged, the lower the pressure was, and it was linear. It was very clear. So after all of these efforts to try to clog it, which by the way, the lack of being able to clog it and the lack of being able to get to a high PSI was really encouraging by itself as far as putting a sorter in the BSL-3 is concerned. But we still really wanted to try to do this test. So we decided to artificially, increase the pressure of the system without there being a clog. And you can do that manually. So we adjusted the pressure to the maximum that the instrument would allow before it automatically turns off, which is approximately 14.14 psi. And that converts to, I think it's 975 HPA, either 950 or 975. Above that, and the instrument will just turn itself off. So that's another built-in safety feature of the Tyto. So once we made that decision, we moved on to figuring out if there was a volume of sample that hit the mixing propeller, so that, or the agitator, so that the spray or splashing caused by the mixing was at its highest. And it was interesting because instead of finding a lot of spraying or splashing, you can see here, we use this red dye and running buffer to try to find spraying, that there was none. There really wasn't. And the reason for that is you can't see from this angle, but if you look down from the top, the way that the mixing propeller is designed, which is a great design, I've really come to appreciate it since this test, is it pulls the liquid inward. It creates basically a funnel inward. It doesn't push the liquid out. So it really has almost, not basically, there was no spray that we could find. At about a sample volume of 450 and 850 microliters, we did detect what we're calling microbubbles, which were just these really tiny bubbles in the solution, and they would kind of flow and then they would be pulled in. It was not really a concern, but it was the most that we could like perturb the sample. So we decided to run at least 1 mil of sample through the system and run it all the way down to the bottom, so we could test sample at its maximum, disturbance. Okay. So to sample the interior of the cartridge, this took a little bit of creativity. We ended up creating a system where we glued a pipette tip, a very skinny pipette tip, to the lure lock, and then we dipped the pipette tip into a mounting media that we make here at Tulane. And the mounting media doesn't drip. It's not so viscous. Like, it's good enough that we can dip the pipette tip into it as far as viscosity goes, but it's viscous enough that it also didn't drip into the sample. So it was interesting to try to find the right material for this, and also had to be clear so it could be put on a slide. So basically, once we had this pipette tip that was dipped into the mounting media, we attached it into all 3 chambers, not just this chamber. We attached it, and then we ran the sample through the instrument, and any aerosols that were created would hopefully stick to that mounting media. We also tested that the aerosols did stick to mounting media with a spray bottle, and they did. And then we could pull out the pipette tip and rub the tip over a slide, and then look at that slide in the microscope. So this was the system for testing the inside, all three chambers of the cartridges. To test the exterior of the cartridge, this is more along the lines of what you do for conventional cell sorting. So we use the cyclic D cassettes, and we ran the samples for 10 minutes, like they do also for conventional sorting. And we tested 2 locations above the cartridge. So this one location here above 2 of those filters, and then a second location above the 3rd filter here. So there were 3 interior samples that we took, 2 exterior samples that we took. And then we also did another sampling outside of the closed sort chamber door and this vent here that I mentioned earlier. The results were encouraging, if not exciting. I mean, exciting in some senses and boring in others, but we detected no aerosols of any size. We tested all 3 sizes with this method, the 50 nanometers, 100 nanometers, and 1 micron beads. We didn't detect them at any size from inside the positive or the negative chambers. We weren't able to, in the end, so far, we haven't been able to make a determination for the input chamber, and I'll explain why in the next slide, but we didn't detect any in the positive and negative chambers. So that's like the pipette test, the pipette tip. We were also unable to detect beads of any size outside of the cartridge, and we were not able to detect them outside of the instrument. So that was very encouraging. We have some work left to do. One is to determine the lower limit of detection. We know that after looking at the slides for hours that we were not able to see any beads. But we're working on ensuring that we're looking at the right plane, the right focal plane in the microscope, so that we're definitely not missing any of those 50 nanometer beads because they're hard to see. So we're very confident that if aerosols are generated, it's very few because we couldn't detect any, but we still need to go back and create that threshold, that lower limit. In addition, we have to optimize our bead loading system because when we loaded the beads, we accidentally touched the sides unknowingly of the lure lock. And so when we took the pipette tips out, they brushed against the sides and contaminated the sample. So we're working on a funnel system to add the beads without touching the sides, and then we'll be able to get those results. We need to repeat this for multiple lots and also test the high-speed cartridges. This should be really interesting because they do operate at a higher pressure than the regular cartridges. If we do detect beads, say in the input chamber, aerosols in the input chamber, or after we do more replicates, we detect beads in one of them, our plan is to think about adding a layer of mineral oil to the top, let's see, of the cartridge, go way back, like here, such that, or even the top of the sorter chamber, wherever we need it, if we need it, would be to add a layer of mineral oil to prevent aerosols from escaping through the top, which I think is a really interesting idea. And that would also create a safer sorting scenario if we had to. So I just wanted to give a big thank you to Megan Varnado and Caitlin Didier. They are specialists in my flow core and our flow core at Tulane. They did a lot of this work. We have had, as I mentioned in the first slide, it's been a journey trying to, we thought in the beginning that this would be very, very easy, and it wasn't. It was, like I said, like impossible to clog, impossible to get the pressure high, lots of difficulties making this thing break. It does not break easily. And they have been very patient, and we've tried so many things to get to this place. So big thank you to both of them for their persistence. And then from Miltenyi, Daryl, Megan, Ellie, Elaine, Matt, Dewitt, Crystal, extremely helpful, extremely knowledgeable. And I'm sure my conversations with them will be continuing as we continue testing. And my boss, Mitty Kaur from Tulane, and Cecily in the confocal core, who made the proprietary mounting media that we were able to use in pipette tips, because all of the commercial ones that we tested did not work. They all basically dripped into the sample. And then just here, this is not a very good picture, but this is our new Tyto in the BSL-3. And we're not allowed to have cameras in there or cell phones in there. So it's a picture taken from outside the BSL-3 actually, but we are very excited to start working on it in this space. So thank you for your time.